Hello everyone. I am here, Armanti, and I would like to say many things as I and my team get this opportunity to present our research here in this conference. And my team, they are Bapak Profesor Amran Laga and also Ibu Dr. Andi Hasisa. I thank them for their support. And our research entitled Screening Marine Lactic Acid Bacteria of Brown Seaweed Sargassum Pond System Symbiotic, producing bioactive compound from Indonesian waters. So that's the title of this research. And well, we will move to the main presentation. Well, here I am sharing the presentation. And as I mentioned before that, our research here entitled Screening Marine Lactic Acid Bacteria of Brown Seaweed Sargassum Pond System Symbiotic, producing a bioactive compound from Indonesian waters. As introduction here, we're talking about primary and secondary metabolites. As we understand that the potential of Indonesian marine resources includes fish and non-fish biological resource. And here, seaweed is a very important biological resource. They are the largest producer biomass in the marine environment. Well, we're also talking about symbiont bacteria. So the microbes can be present as a living symbiont in association with various algae in the form of epiphytes or endophytes. So epiphyte is a bacteria that grow in the surface of the algae or other marine organism, while the endophytes, this symbiont bacteria grow inside of the cell of the organism or seaweed. Well, symbiotic bacteria are involved in the production of metabolites related to bioactive compounds from their host. And talking about antibacterials, brown algae and other species have been extensively analyzed for their antibacterial and antifungal activity, but here we're not talking about antibacterial from the brown algae itself, but we're talking about antibacterial from their symbiont bacteria. And we also need to we also need to inform the authors that some research already published and mentioned like bacteria of marine strain may have better potential than their terrestrial counterparts. This is because of the essential nutrients provided by marine biotopes to maintain this bacteria and its extreme environmental niches. And marine strain and their byproducts may have potential value in the food processing, fermentation, pharmaceuticals, and biopolymers industries. And this is some of the reason why we want to more focus on analyze the symbiont bacteria of this seaweed. Okay, as purpose here, we have three main purposes. So the first purpose is the bacteria isolation. So we want to isolate this bacteria from seaweed and based on the laboratory standard procedures, of course. And the second purpose is a paper disc diffusion test because we want to analyze the bioactive compound that produced by the symbiont bacteria. And the third purpose is bacteria identification because we want to identify what kind of type or species of this symbiont bacteria, and that's very important. As methodology here, we have equipments and also materials, and equipments here that we use generally based on the standard laboratory equipments, for example, like petri discs, reaction tubes, Erlenmeyer's, Baker glasses, and measuring glasses until microscope, and etc. And for the materials were used conducted this research include brown algae, it is sargassum polysystem, pure culture bacteria, Staphylococcus aureus and E. coli. They are gram-positive and gram-negative pathogenic bacteria. And some medium like nutrient growth, PCA, MHB, MHA. We also need sterile water, sterile seawater, 70% and 95% of alcohol. And we also need some materials for cell staining, for example, like for gram staining, spore staining, and also Z-Nielsen staining. Well, the next is the procedures. So we have here 4 main procedures. And I'm sorry, I didn't type the main title of this procedure, but here in the first procedures, they are sampling. It is sampling. And the location of sample derived was in Lima Island, and it was in Banten, where specifically brown seaweed and also other wild seaweed are grown, and they are massively grown there. And sample collected about 100 grams. So it is the sample is a sargassum polysystem, and sample maintained in fresh seawater with oxygen addition for further laboratory analysis in less than 24 hours. So this oxygen addition is to maintain their natural habitat for this seaweed, and also the bacteria that live inside the seaweed or the surface of the seaweed. Well, the second procedures, it is the isolation procedures, isolation step. So isolation was started by enrichment sample from the algae. The homogeneous sample was inserted into a 30 mg medium, shaken at room temperature for 24 hours, and then sample diluted with a 9 mg until 10-5 dilution. So we need 5 dilution. And each dilution was grown on an MHA medium, so agar medium, and then we incubate them in a specific temperature and also time. And after that, we hope that we can produce, and they can produce like a colonies that show inhibition zone. So colonies of bacteria that produce antimicrobial compounds were characterized by a clear zone that appear around the colony. And the third procedure here is the agar diffusion test. So testing of bioactive compounds of symbiont bacteria was carried out by the agar diffusion method. So we prepare like paper disc containing the supernatant and also negative control medium without addition of symbiont bacteria, and also we have a positive control, which is a chloramphenicol, and they place on the surface of agar medium containing tested bacteria. It was pathogenic gram-positive bacteria and negative bacteria. And after that, we incubate them and observe the zone inhibition, and also we measure the zone inhibition. And after that, these chosen bacteria, the bacteria that produce bioactive compounds, then we identify them again. So the isolate was observed morphologically to determine the type of symbiont bacteria, and also we do the DNA test to see the species of the symbiont bacteria. So we have here result in discussion. And as first result, it is bacteria isolation result. And as we can see in the pictures, we have here the colonies that already grow, and some colonies produce what we call as an inhibition zone. So the individual microbial cell multiply so rapidly that within 18 to 24 hours, a feasible mass of cell is formed and is called a colony. So the ground sample consisted of as many as 5 petri dishes, resulting in some colonies that show an inhibition zone. However, only one colony was possible to be isolated and has the clearest zone of inhibition. The result of the identification of chosen colony growing in mixed culture show a round shape, white color, flat edges, and has convex, shiny elevation. So this is the sample of the symbiont bacteria, the colony that produce zone inhibition, and it is very clear as we can see in the pictures. And then we have here the second result, paper dish diffusion test. And application of antibacterial compounds produced by isolates of symbiont bacteria showed different inhibitory activities against the two bacteria tested. These tested bacteria are gram-positive and gram-negative pathogenic bacteria. And we observe the inhibition zone around the paper dish. And paper dish with the supernatant applied to plates containing gram-positive bacteria show a stable, a clear zone even after an incubation period of 48 hours. But as for gram-negative bacteria, the surrounding paper dish show inhibitory activity but gradually became cloudy before the incubation period reached 24 hours. So we're talking about here the supernatant that we apply into the gram-positive and gram-negative bacteria. And here the result, as we can see, that the supernatant is more effective in inhibit the growth of gram-positive pathogenic bacteria, Staphylococcus aureus, based on the result as we can see in the pictures. And the next is here we show a table inhibition zone diameter results. So positive control had wider spectrum in inhibiting both types of test bacteria. So the positive control here, it is a chloramphenicol, and they work very good in both gram-positive and gram-negative bacteria. But here the negative control, it is the medium growth without inoculation of symbiont bacteria, showed no activity or no zone inhibition. So it's pure the supernatant work effective to the tested bacteria. So in general, from the measured stability of the inhibition zone, the antibacterial properties of the supernatant produced by symbiotic bacteria work as a bactericidal against gram-positive bacteria. And only bacteriostatic on gram-negative, but we can say here bacteriostatic on gram-negative, but we're not sure about that because we cannot measure are there any form of inhibition zone before reach the time of 24 hours of incubation. So yeah, we only see like a cloudy surrounding of the paper dish. So maybe some like they try their best to against the gram-negative, but they didn't work. Well, result and discussion. For the third result and discussion here, we have identification of symbiont bacteria. And for the symbiont bacteria, some types of observation we did for the colony, and also we also observed the cell. The cell with the cell staining, for example, like gram staining, spore staining, and also Z. Nelson staining. And also we also did the biochemical test, and of course the last test is a genetic test or DNA test. And here, characteristic that we have been known from the identification of symbiotic bacteria microscopically and biochemical and with biochemical tests include they are rod shaped and not acid fast, not forming spurs, not motile, and also they are aerobic growth, negative catalase, and positive carbohydrate tests. And the result of the alignment of DNA sequence encoding 16S rDNA isolate of symbiont bacteria show an accurate score for the species level with 99% similarity of the sequence in gene bank. And it is known that the species homology of the tested isolate was Lactobacillus plantarum. And the classification of the bacteria isolate is bacteria, Firmicutes, bacilli, lactobacilli, lactobacillus, and of course the species is Lactobacillus plantarum. Well, as conclusion here, the results of the isolation of symbiont bacteria obtained several isolates. Well, but while the colonies that produce the best inhibition zone and possible to be isolated from mixed culture were one isolate, so only one isolate that possible to be isolated. And the result of the paper diffusion test showed that the isolate could inhibit the gram-positive bacteria Staphylococcus aureus. And through molecular testing, DNA, it was found that the species of the symbiotic bacteria, Sargassum polycystem, was Lactobacillus plantarum. So the types of LAB strain obtained from the marine environment show positive potential in their utilization. So we really hope further research will open up opportunities for the LAB potential of marine strain in the future. So yeah, this is what we really hope we can utilize this kind of lactic acid bacteria marine strain. Well, I would say that this is the end of my presentation, and in the end of my slides, I show you the pictures of Sargassum pond system, and they are so beautiful. And they grow around our beaches, and may you can find them as well. And I also I hope that this presentation and also our other presentation can give benefit to others or community who needs this kind of information. So thank you for your kind attention, and we'll see you next time if God willing. Thank you so much.